Good afternoon, everyone, and thank you very much for joining us for today's Richard Dole seminar, which is the last for the term. Just a reminder that this lecture is being recorded and you can access it again via WebLearn. Not all lectures are made publicly available, but this one will likely be. But just to be safe, always good to treat all information delivered during the seminars as confidential and not to share it with any other parties. We'd love to hear any questions that you might have, and you may notice that we're back into the live Teams version. So it'd be great if you can send this into the question and answer section, and only the presenter will have access to this. We won't be publishing this anywhere. It would also be good if you can tell us about any technical difficulties using this section. Today we're pleased to have Professor Angela Brueggemann, who is a professor of infectious disease epidemiology here at the University of Oxford. Professor Brueggemann has a long-standing interest in assessing the impact of vaccination and antimicrobial resistance, and regularly advises bodies like the World Health Organization in this area. Amongst other projects, Angela's efforts are now focused on the IRIS initiative, which we're going to be hearing about shortly. The title of Professor Brueggemann's talk is The Irish Initiative Investigates Changes in Invasive Disease in 26 Countries During the COVID-19 Pandemic. Angela, I'll now hand the floor over to you.
That's great. Thank you very much, Karen. Thank you for the invitation to come and give this talk, and also for that introduction. When I first came to Oxford in 2000, I came as a DPhil student, and I joined Green College, which is now called, of course, Green Templeton. And Richard Dole, as many of you will know, was the founding fellow of Green College, and indeed was the first warden of Green College. So I met him a number of times when I was at college, and I never failed to be impressed with Richard. We'd have chats, at dinner or whatever. The last time that I remember speaking to him was at a garden party, I think the summer of 2004, so probably around a year or so before he died. And my now husband, Matthias, and I had a long chat with Richard in the garden, which was lovely. And I don't remember all the details of what we spoke about, but I do remember him being very approachable. And I think as a student, I was probably rather overawed by him, quite honestly. And so now, fast forward a number of years, to actually be working in NDPH and indeed give a lecture in the lecture series, sort of in his honor, is quite special in this context. So thank you very much for that. So you can see the title of the talk. So this talk will be about the IRIS initiative, and the motivation for setting up this initiative was the pandemic. So we're all very well aware now of the past year and a half or so, and it was about early April, I think, when we started having some discussions, a small group of us who do work in pneumococcal disease and other similar invasive diseases, which I'll explain in a minute. We were having discussions around what the COVID pandemic might mean for invasive bacterial disease. And the reason we were having that discussion was because it's well described and well recognized that there can be invasive bacterial infections which occur subsequent to viral infections, particularly lung infections. So, for example, influenza followed by a subsequent pneumococcal infection is not uncommon, and indeed in the 1918 influenza pandemic, the subsequent pneumococcal disease contributed significantly to the morbidity and mortality of that pandemic. So we were having discussions around whether this might be a similar situation. In other words, a respiratory virus, SARS-CoV, might indeed lead to increased invasive bacterial disease. But equally, countries were taking measures to try to stop the spread of SARS-CoV, and so that could indeed lead to decreased invasive bacterial disease, because the organisms we're interested in are also transmitted through the respiratory route. So we might indeed see a decrease in disease. And then whatever happens, we may well see that these disease perturbations fluctuate, I guess, as the measures are lifted, the containment measures are lifted. So these were the discussions that we were having. And as a result, we then set up this study. Before I get to the details of the study, there will be some of you who aren't familiar with these bacterial organisms. So let me just give you a few minutes of biology. So the main focus of our work is Streptococcus pneumoniae, which we also call the pneumococcus, and I'll probably typically refer to it as the pneumococcus. Also Haemophilus influenzae. Many of you will have heard of Hib, Haemophilus influenzae serotype B. And then Neisseria meningitidis, which we also refer to as the meningococcus. And these three organisms are leading causes of invasive diseases such as meningitis, septicemia, and pneumonia, which is severe pneumonia, that is bacteremic pneumonia. And these are all life-threatening disease. By invasive disease, we mean that the bacterial organism, or indeed any microbe, has invaded a normally sterile site, such as the CSF or the blood. So these three organisms are leading causes of these types of diseases. And the age groups who are most at risk are the children who are less than 5 years of age, and also the older adults, typically those over the age of 65 years. But what's important to understand about these organisms is that the ecological niche for these organisms is indeed the healthy child. So the nasopharynx and for the meningococcus, typically the throat of children, that's where these organisms reside. And therefore, they're all transmitted via respiratory secretions, which is relevant to this work. If we consider the morbidity and mortality associated with these diseases, then we can consider, first of all, pneumonia, which is a leading cause of death globally. And the most common causes of pneumonia are pneumococcus and then also Haemophilus influenzae. There was a study that was done in 2016 called the Global Burden of Disease Study, which looked at a number of different types of diseases. And I've just extracted one of their maps from that study. These data are for children less than 5 years of age with pneumonia, and it's the mortality rate per 100,000 people. And you can see the different colors indicate the different mortality rates. So in much of Europe and most of North America, Australia, the rates of disease are relatively low, about 4 cases per 100,000. But you can see that much of the world is green, meaning that we're looking at rates of disease of somewhere between 10 and say 50 cases per 100,000. And then if you look at parts of Africa, India, and so on, then disease burden starts to become quite high indeed. Anything over 100, even up to 500 cases, deaths per 100,000 population due to pneumonia. If we focus just on pneumococcus, which is the predominant cause of that pneumonia, then from this study in 2016, there were an estimated nearly 200 million episodes of pneumonia, which led to over 1 million deaths worldwide. And if we put that into context with the current COVID pandemic, so in the first year of COVID, there were 118 million cases and over 2.6 million deaths worldwide. And this isn't meant to be a comparison or competition of grim statistics. It's just to put into context that pneumococcus is also a major, not just burden of disease, but also of mortality. And the data here, of course, from 2016 are 5 years ago. If we consider meningitis, meningitis is a disease which is more rare, relatively speaking, but which is quite devastating as a disease. So again, from this 2016 Global Burden of Disease study, this is one of their graphs which shows the age standardized incidence of meningitis per 100,000. And you can see that most of the world is blue or shades of blue, which are the low rates, somewhere between 0 and say about 20 cases per 100,000. But again, in Africa, the burden of disease is much higher, upwards of 200 cases per 100,000 population. And many of you will recognize that this is what's called the African meningitis belt, because disease burden is much higher here. And we know, in fact, that much of these reports of meningitis are indeed underestimating the burden of meningitis for various reasons we could go into later. So from this study, then, the number of deaths due to meningitis across all age groups was about 300,000 in 2016, from an estimated nearly 3 million meningitis episodes. And what's important probably to understand about meningitis is that the case fatality rate is very high. Now, this varies depending on the organism that's causing meningitis and indeed the speed at which, well, treatment and the speed at which that treatment is implemented, but could be upwards of 70% without any treatment. Those who survive meningitis are often left with long-term disabilities, often hearing loss or developmental disabilities in young children, and so on. So it is a very devastating disease and is a major invasive disease globally. Now, our interest is in those 3 pathogens, but I'm also going to talk about Streptococcus agalactiae, which is also known as Group B streptococci or just GBS. So Group B streptococci are also a leading cause of life-threatening invasive disease, but in particular neonatal disease. The reason we're including it here is because it's not a respiratory pathogen. It is normally carried in the healthy female gastrointestinal and lower genital tract, and therefore the risk is to both a pregnant mum and to her child, and the risk of transmitting group B strep during labor and delivery, and that potentially infecting the child. So it is a major cause of a particular type of disease. It is increasing amongst older adults, reasons for which are not entirely clear. So it's a major cause of invasive disease, but in the context of this IRIS program, it's not transmitted through the respiratory route. So to address the questions that we had around increases in invasive disease or potentially decreases as a result of the COVID pandemic, we set up the Invasive Respiratory Infection Surveillance Initiative, or the IRIS initiative. And the aim was very simple: to, as quickly as possible, assess the changes in the incidence of invasive disease due to these pathogens I just described. And we did that by assessing the number of cases that were submitted to national reference laboratories as part of their routine surveillance program. So these data were being collected by national reference laboratories already, and we were able to pull laboratories into this study and then combine these data. So things happen very quickly in a pandemic, and many of you have been involved in setting up studies rapidly, more rapidly than most of us are used to setting up studies, and IRIS is no exception. So in March of last year, the WHO declared the COVID pandemic. We started having these discussions sort of early April, and in the span of about 6 weeks' time, recruited 37 laboratories across the world. We've collected data from the start, and that data collection is indeed ongoing. This is very much a prospective study that I would envisage lasting some years into the future. We were able to post our first manuscript to medRxiv in November last year, and we've just published the first paper in Lancet Digital Health a couple of weeks ago. So I said this was an international network of laboratories. You can see them all here. Many of them, of course, are in Europe. Again, we were tapping into our network of collaborators, long-standing collaborators. So we had decided we would set up this study, and so we contacted people that we knew running these laboratories to invite them to join. And we had a very enthusiastic response. There were only 3 countries that said they weren't able to join, but that was only because their data were indeed disrupted during that period of time, and they didn't feel they could provide high quality data for last year. But you'll see we have quite a few laboratories involved from Europe, but then Canada, in the US, in North America, Brazil, South Africa, and New Zealand, and then China, Hong Kong, and South Korea, and also Israel. So just for reference, we have 3 Southern Hemisphere countries, and the rest are all in the Northern Hemisphere. From the start, we also intended to try to keep things as simple as possible. So all the laboratories were really stretched in terms of COVID testing. Some were actually doing COVID testing. Others had staff that were seconded to other labs to do the COVID testing. So laboratories were generally fairly stretched. And so we wanted to keep things as simple as possible to try to get at good quality data with minimal effort. So we set up a private IRIS database using the PubMLST platform and infrastructure. I'll describe that in the next slide. And we only requested minimal data to start, so cases of invasive disease and the date of sampling, and then we could follow with data updates later. Indeed, many laboratories did provide more data at the start, but the rest of the data are still relatively incomplete, but we're working on that. So we first looked just at cases and date of sampling, and then the database is private, but IRIS participants can log in and they can enter their data themselves, or they can send it to us and we can upload the data. So many of you are probably not familiar with PubMLST. So MLST stands for multilocus sequence typing, and it's a genotyping technique which was developed a bit more than 20 years ago by Martin Maiden in zoology and Brian Spratt, who used to be here at Oxford. And it was a way of genotyping bacterial organisms. It is the gold standard way of genotyping organisms. And from its inception, it was based on sequence data, and therefore those data are easily portable. And so the idea was to not only have a robust typing method, but also be able to share that information very easily via the internet. So PubMLST is the public platform for MLST, and Martin Maiden and Keith Jolly run the platform and servers in zoology. And Martin and Keith and I hold a Wellcome Trust grant, which funds this PubMLST infrastructure and the databases and so on. So in PubMLST, there are now 129 databases, all of which are open access for different microbes, predominantly bacterial microbes, but some yeast and so on as well. We have an international team of curators who consider the data that's put into PubMLST. And whilst it initially started off as just short fragments of sequence, we have over the past few years been adding genome data to PubMLST as well, and there are now nearly 620,000 assembled genomes with corresponding metadata that are all freely available in PubMLST. In addition, there are tools for analyses and curation of genes and specific loci of interest as part of PubMLST. So if you go to a particular web page, this is the pneumococcal web page, then users can access the sequence data, they can access the isolate collection, and they can access genomes. So we use this platform to set up the IRIS web page. This is different because it's a private database, so it uses the same infrastructure. It's just that the only people who can access these data are indeed the IRIS participants through a login and password. And so we have one each database each set up for each of the bacterial organisms. So it's very important for these laboratories that the data remain private, at least in terms of specific data that might identify patients. So there is indeed no identifiable patient data included in IRIS. However, in countries where they have, say, a small number of cases of meningitis, it could be possible with a bit of information. If you knew that a case had been sampled on a particular day and it was a case of meningitis, you might be able to identify that particular person. So the data remain private, and this is important because these are reference laboratories. However, the laboratory that submits the data to IRIS retains ownership of their own data. They continue to use that data as they wish and publish as they wish, but they're also part of this consortium. So we, as I said, recruited 37 laboratories from 26 countries and territories, and those laboratories submitted data for one or more pathogens to IRIS. Now, it's important to remember that these are invasive infections, which are medical emergencies. So these are patients who are treated in hospital and the samples are taken in hospital. It's also important to know that because of the severity of the disease, clinical microbiology labs are generally required by law. And amongst these IRIS laboratories, the majority are indeed required by law to report invasive infections and often submit the isolate to the reference laboratory. The first data analyses that we've performed were on the data up to May last year, so from the 1st of January 2018 through to the end of May 2020. And as I said, we've just published this work in Lancet Digital Health. So what did we find? Well, the first big result was that there was a significant reduction in invasive disease to these pathogens. So I want to mention Melissa Jonsson from Rendsburg. She's a postdoc working in the group. We've been looking at these data, and she's the one who's responsible for the very nice graphs that you'll see over the next few slides to visualize the data that we have. So what we see here are rather simple graphs in that we're looking at cumulative isolates on the y-axis, and then time since the start of year for each of the pathogens of interest. So for pneumococcus, we have over 62,000 isolates, nearly 8,000 isolates of Haemophilus influenzae, and nearly 6,000 of Neisseria. And then we have about 4,000 isolates, just over, of the group E strep data. So you can see that in each curve we have these hashed lines which represent the 2018 and 2019 data, and they look very, very similar, indeed almost superimposable for the pneumococci, similarly for Haemophilus and Neisseria. Obviously changes in numbers there, but the pattern is the same. And then you can see this very clear change in 2020. This red line is marking the week 11 of 2020, which is when the WHO declared the COVID-19 to be a pandemic. So you can see that in each of these three cases, things change right around that time, and that disease, invasive disease reports diminish for these three organisms, whereas you don't see that for the group B streptococci. And the reason we included this was precisely for the reason that we were discussing amongst the IRIS participants about whether or not this could be due to lack of reporting. So were the labs too stretched and not receiving the isolates as they would expect in a normal year? Could that explain this reduction? But the overwhelming sense of that was it was not the case. So laboratories were continuing to receive other isolates as they expected. They didn't see any major drop-off in sampling of these organisms. And indeed, for 9 laboratories that were collecting Group B strep data and also these organisms, they didn't see any difference. So we were confident that what we're seeing, there could be some lack of reporting, but that doesn't explain the result that we were seeing. So we then wanted to understand what was driving this change. If it's not lack of reporting, then what else might be driving the change? Well, of course, countries were implementing different containment measures to try to control the SARS-CoV spread. So we then contacted Thomas Hale and invited him to join as a collaborator, because he's leading a project called the Oxford COVID-19 Government Response Tracker, or OxCGRT, where he's leading a team of over 100 Oxford students and staff around the world, collecting metrics about the types of responses that governments have made for controlling COVID. So they have 20 different indicators, and they're collecting data from over 180 different countries, and they've also recently published this work. We chose the OxCGRT Stringency Index to use in the IRIS study. So in this set of measures, they're looking at 8 different indicators. So when and if countries closed schools or closed workplaces, to what extent public events were cancelled, any restrictions on gathering size, so the number of people that could gather, whether public transport had been closed, what the stay-at-home requirements were leading up to full lockdown, and then any restrictions on movement either internally within a country or indeed between countries, international travel. So they calculated each of these on a range of scores, and then they had a simple algorithm which converted that to a stringency index, which ranged from 0 to 100. So 100 is the most strict policies, and in this map are colored blue, and as you'll see on subsequent slides. So the blue lines are more strict, more stringent policies in countries. So I've pulled the data from last year on the 31st of March, 2020, and you'll see that pretty much across the globe, everyone was in fairly stringent or highly stringent measures, with a few exceptions, but mostly the world looks pretty blue, as in very strict containment measures. If we fast forward a year, then on the 31st of March 2021, you can see that things have changed. So this was 2020 data. Now things are starting to open up, and the stringency is more in the sort of mid-range for many countries. Certainly there's still, UK included, quite strict measures in place, but other countries are relaxing those measures. And this will start to become very important for us as we follow the data over time, our data over time. So in order to try to use these data in combination with our invasive disease data, Melissa created these clever graphs where we have, again, cumulative curves, and now we're just looking at pneumococcal data. And I'm just showing you 4 countries at the moment. I'll show you all countries in the next slide, but just so you can see these. We have the same sort of cumulative curve. So we've got 2018, 2019 data in hashed lines, and then we've got this red line, which is the 2020 data. And we're looking at number of isolates across the week of the year. And here we've just got 4 countries, the English data, just over 12,000 isolates. And then superimposed in the background, week by week, is the mean weekly OxCGRT stringency value on a scale of 0 to 100, the same that you just saw. So what you can quickly see is that as the containment measures started to become more strict, we start to see a change in the number of invasive cases that have been reported. It's worth noting, while you can see it more clearly here, that you can see those changes occurring even amongst countries for which the stringency was not as high as, say, we experienced here in England. But in Finland, they were in sort of moderate containment measures. And indeed, Finland, Denmark, Iceland, and Sweden all looked actually quite similar in terms of the stringency of their measures. And the resulting reduction in invasive disease was consistent across all those countries. So if we move forward, you can see all of the countries here. I realize it's small, but what I'd like you to take away from this is that the patterns look really quite similar. So this is now the invasive pneumococcal data per country for each country that's involved in IRIS. And I think the key take-home message here is, first of all, that the stringency levels did indeed vary by country. You can see Finland, Iceland, Sweden all have moderate levels of containment, as compared to the 4 nations within the UK. And yet there is still this sustained and quite dramatic reduction in invasive disease in all countries. These data describe the pneumococcal data, but we have in the paper graphs for the other organisms as well. And so you can see that for Haemophilus and Meningococcus, we see the same pattern across all 3 pathogens. So the discussions that we were then having within the group at this stage were, well, so stringency clearly matters, or the containment measures clearly matter. That seems to drive some of the change, but there are clear variations in terms of the level of stringency among different countries. So how else might we explain the reduction in disease, even if containment measures are not as strict in one country versus another? And we had discussions around the mobility of people, and that despite what governments may or may not instruct us to do, that people do make their own choices and assess their own risks. So Google produced started early last year, these Google COVID-19 community mobility reports, where they aggregate and anonymize the location history data from people's mobile phones. And they do that in 6 different categories. So they're assessing the movement of people in 6 categories. You can see them listed here. We were particularly interested in the residential and workplace data. So what they do, and you can access all these data online on their website, is they calculate a daily mobility data measure for each country, and that's the percentage change from the baseline day, and they define the baseline day as the median value in the first 5 weeks of last year, and then the percentage change from that baseline day. It's important to note that there are no data for China because Google is censored in China, and neither is there data for Iceland because Iceland is a small population, and therefore there's the risk of identifying individuals within that population using these data. Also note that the UK data are not separated into 4 nations within the UK, but aggregated as one data set. So Melissa again worked her magic and depicted 2 different types of data on these graphs. What you see on, this is one graph per country, as I say, UK aggregated. So what you see in each of these graphs in blue is the residential data. So this is the movement to and from residences, and conversely to and from workplaces in red. The reason they're spiky like this is because Google is collecting daily data. So at the weekends, you get a data spike, and then when there's a holiday, you get a spike. So around this period of time is the Easter weekend. So we see big spikes there. And then the dashed line indicates, again, week 11, when the WHO declared the pandemic. And I think it's relatively easy to say that, again, there's a lot of similarity amongst the countries. So basically you have very little, apart from some peaks, relatively little change before the pandemic is declared, and yet you see differences then after the pandemic is declared, as in people are spending more time at home and less time in the workplace. And that's pretty consistent across every country with some variation in terms of the magnitude of that difference. But people were making their own choices about how much risk they were prepared to take in terms of mobility, was our interpretation of these data. So human behavior is also very important in terms of what people do in the midst of containment measures in the midst of a pandemic. So we then were able to use these data to start to quantify the effect of the containment measures on the weekly number of cases. So this work was led by Noel McCarthy and David Shaw. Noel was at Oxford some years ago. He did his DPhil here. He's now a professor of population health medicine at Trinity College in Dublin. And many of you will know David. He was a master's student in our department last year, and he's now working as a DPhil student in my group. So they led the statistical analyses where we were attempting to quantify the effect of the containment measures on these numbers of cases of invasive disease. And the measure was after trying to assess what was happening after the containment measures were put in place. So assessing whether there was evidence for both a step decline or just step change in cases per week, and then a slope subsequent to that. Because we have a lot of pneumococcal data, by far and away the greatest number of isolates, then they also performed a meta-analysis to summarize the changes that occurred 4 weeks and 8 weeks after the interruption. So Noel describes it like this, a simple graph where we have time over the x-axis and then some outcome on the y-axis. And so we have our slope or disease pre-intervention. Then some sort of intervention is introduced. And in our case, we're looking at when the mobility, using the Google mobility data, when the mobility of people changed. And then that results in a step change and then a new slope subsequent to that. So they calculated the interruption time point using that Google mobility data, and they selected the week which contained the day that was halfway from the baseline to this new lower minimum value. You'll recall that I said that both Iceland and China, we didn't have mobility data for those, so they used the modal week of other European countries for the Icelandic calculations. And for China, they looked at news reports of policy decisions to make a choice. And so they used weeks 5, 7, and 8 for China, Hong Kong, and South Korea, respectively, as the week in which to assess that intervention. For New Zealand, it was week 13, and for all other countries, it was either week 11 or week 12, bearing in mind that week 11 was when the WHO declared the pandemic. And they also controlled in the models for seasonal variation, the Northern Hemisphere versus the Southern Hemisphere. They combined the data across the countries for all 3 pathogens, that is the Pneumococcus, Haemophilus, and Neisseria data. And they limited those analyses to the countries who could provide, to the countries who provided data for all 3 organisms. And then they focused on the pneumococcus because we have over 62,000 cases of invasive pneumococci, and they looked at country level analyses to assess the patterns across the different countries, and then looked at combined analyses to assess the effect of interventions that might have varied across countries. You'll recall in the very beginning when I was describing the biology that I said that these organisms, these 3 organisms, are largely carried in healthy children. So school closures could have played a major role in the reduction of disease because the children were no longer within school and exchanging these organisms. So they assessed the effect of school closures measured based on the CGRT metrics. So what did they find? Well, they estimated that these social changes, the mobility changes caused by the pandemic, led to a 30% decrease in the incidence of pneumococcal invasive disease shortly after that step change, with a subsequent 13% additional weekly reduction on average up to the end of last May. So that's the slope change from that graph. What was clear is that there were similar patterns across the world regions, across the different countries, and indeed there was no evidence for any latitudinal changes in terms of hemispheres, which you could see from the graphs that I just showed you, but now it was quantifiable. And the school closures did not significantly improve the model fit, so we couldn't explain the reduction simply based on school closures. And finally, in terms of statistical analyses, there are other data in the paper, but just to show 2 final forest plots that they were then able to, Noel and David were able to assess the country-specific incident rate ratios after this interruption in mobility. And this is again for the pneumococcal data. So you can see the countries listed here, incident rate ratio and the weights. And 4 weeks after the change in mobility, the incidence of invasive disease was decreased by 68%, and 8 weeks it had decreased by 82%. So that was a way to quantify what we had been seeing and observing using the previous metrics. So where do we go from here? Well, there's no evidence at the moment yet for any return to normal, that being what we might expect in a normal year for invasive disease. Mark van der Linde runs the streptococcal reference unit in Germany, and he's one of the key people that was involved in setting up IRIS. And indeed, it was discussions that we were having with Mark which led to the setup of IRIS. So Mark said it was fine to show the German data. So what you see here is a graph similar to what I showed you before, where you can see for pneumococcus, these data, the blue and the orange, are for 2018 and 2019. And throughout 2020, there remained the sustained reduction. For pneumococcus, the same is true for Haemophilus and Neisseria, and indeed in 2021 things haven't really changed. So the pneumococcus, Haemophilus, and Neisseria data remain persistently low, and the GBS data remains more or less as expected per year. These are German data, but we see this pattern across the countries. So what are the findings from IRIS to date? So first of all the countries and territories in IRIS, all 26 of them, experienced this significant reduction in invasive disease due to these 3 pathogens, and that was sustained over time, certainly within our first tranche of data analyses, but continues to be a relatively sustained reduction, and we're continuing to monitor for changes as they might occur. but we don't see any significant changes in group B streptococci. So we do think that reporting is proceeding more or less as normal for these organisms. And what was interesting is that we did see similar trends in terms of this reduction of disease, despite the different levels of containment measures, so different strictness or stringency in these control policies. And this could be quantifiable in terms of this reduction of 68% at 4 weeks and 83% at 8 weeks. And indeed, this equates to about 6,000 fewer cases of invasive disease due to those 3 organisms across these 26 countries. So overall, the containment measures for COVID were, of course, designed to reduce the spread and transmission of SARS-CoV-2. So the containment measures plus the public information campaigns. But it also led to, we think, the most obvious explanation here is that also led to a reduced transmission of these 3 bacterial pathogens, which are also carried in the respiratory tract and are transmitted through the respiratory route. And that reduced transmission led to a significant reduction in this life-threatening invasive disease in many countries worldwide. And although we've not measured it across other countries, we would expect similar reductions in other places as well, given that there is this reduced transmission of these organisms. And what's, I think, especially important is that we're obviously very focused on COVID vaccinations and so on, and rightly so. But it's really important to stress that there are vaccines that already exist for these organisms as well, Pneumococcus, Haemophilus, and Neisseria. So I've shown you data in the beginning of the talk that demonstrates just how much of an impact these organisms make to global morbidity and mortality. And we have safe and effective vaccines already for these organisms. Yes, those vaccines are implemented in many countries, including the UK. But the use of those vaccines in national immunization programs is far from comprehensive. So we need to maintain a focus on vaccinating young children, which is what these vaccines are designed for, vaccinating young children to prevent the spread of these organisms and also prevent disease. So then in summary, the IRIS initiative was very quickly established in response to the pandemic. And I think what has been most rewarding from this is that it was very quickly an international network and very much a partnership. There's a lot of enthusiasm from the laboratories to being part of IRIS and sharing the data, and a real sense of cohesiveness and collaborative efforts in terms of setting up IRIS and sharing these data. And one of the reasons that we were able to do this so quickly is because these surveillance programs already existed at a high level. So these laboratories, the majority of them indeed have been doing surveillance for several decades, some is 50 years or more. And so the quality of the data coming out of these laboratories is already high. The surveillance programs are well established, and we were able to maximize that in this study to include those data in IRIS. And finally, just to say that PubMST as a platform made this very easy to set up. So we were able to set up a database. Keith set up that database very quickly. We could start adding data and analyzing those data from within PubMST more or less straight away. So with respect to funding, we have a long list of funders in our paper because each of the laboratories have various sources of funding, but I would like to recognise the Wellcome Trust. I've got a Wellcome Trust Investigator Award, which part funds this project, and Martin and Keith and I have a Wellcome Trust Biomedical Resource Grant, which funds PubMST. So I'm grateful to the Wellcome for funding. And I would just like my last slide to be the 95 people who are the co-authors on this first paper, because without all of these people, we definitely wouldn't have had a study, and each has contributed significantly to the IRIS project, but also to the initial paper. So I'll leave it there. Thank you very much, and I'm happy to take some questions.
Thank you very much, Angela. It was a fantastic talk and an incredible amount of work that hopefully can feed into additional future collaboration with everything that you've established in this past year and time before that. If anyone has some questions, please send them through. While we're waiting for some questions, I might kick off. Do you think that given the COVID-19 and the amount of efforts and surveillance that would have required, do you think that some of the changes might be due to maybe underreporting of surveillance of some of these conditions that maybe were disrupted because maybe COVID-19 took over, Or do you think that even if that was the case, would that matter much given the sort of what we know about reducing of person-to-person interaction?
So I think it's a good question, and we've had this discussion as an IRIS group many times. I think that we certainly can't rule out that there wasn't some disruption to the submission of records and isolates to the reference laboratories. But I think that the laboratories themselves feel that any disruption was rather minimal in the grand scheme of things. So they may have received isolates and data later than they would have otherwise done, but they of course are receiving from their local hospitals. And so it might have been that the local hospitals were collecting, sort of batching the isolates and sending them over in a batch, whereas before they had sent them more kind of as they came in. Same for data, there may have been delays in data, but they felt that the overall reporting hadn't been significantly disrupted. And that was the reason for including the Group B strep data, that the Group B strep data is from 9 laboratories, all of whom were submitting the other organisms as well, and they don't see a difference. And then, although we don't show it here, and we haven't analysed it formally, the laboratories were also collecting other organisms besides these, and they would say, well, our E. coli rates are as they were before, or Group A strep, or whatever. So the labs themselves felt that the disruptions were, in their view, probably relatively minimal, and certainly wouldn't have explained the reductions that we see in the way that we see them. So
amazing. So more power to the containment measures. We've just had a question sent in from Rory saying that although there may be reluctance to give vaccines for different conditions at the same time, although of course MMR vaccine does involve it, might the implementation of the SARS-CoV-2 vaccine in low middle income countries provide an opportunity to vaccinate against these other pathogens?
I think it's an excellent question and an excellent point. I think that vaccine programs are going to have to think very carefully about how to, if their vaccination program has been disrupted over the past year, and it will be true for probably lots of vaccination programs that it has been disrupted, even here in the UK, if children weren't being brought to their GPs as they were before. So I think vaccination programs will have to think creatively about how to do that. And certainly if someone is coming for another vaccine, being able to potentially give several vaccines at a time would be one way of approaching that. And certainly there are combinations of vaccines which are given routinely anyway. So I think, yes, that will need to be considered for sure. I think what people have been doing, I've seen several talks over the past year around pneumococcal vaccination. So predominantly it's the young children who receive the pneumococcal conjugate vaccine. Sorry for some technical bits here, but there's also a pneumococcal polysaccharide vaccine, which is given to older adults in many countries. And during the past year, when vaccination programs were disrupted, people were thinking a bit more creatively about how to get people to centres for vaccination. So there was one lecture I heard by someone, I think it was in New York City, where they had a van which was driving around with the childhood vaccinations and going to see the children in their homes to vaccinate them. And there was another story, it was either New Zealand or Australia, but there they had a sort of drive-through centre for pneumococcal vaccine for adults that people didn't even have to get out of their car. They could just drive through and get their pneumococcal vaccine. So I think, yes, I think we're going to have to think creatively about how to do that, because the vaccines are just really essential for these organisms as well as for others.
Yeah. And we've had a question here from Goelette that I might jump onto as well. They've asked about if you think it's possible that lockdown measures also had an impact on the pathogen carriage, and they appreciate that this might not be reflected in lab data. And to add to that, I also wonder if maybe between high and low income countries, if potentially the different measures that maybe were employed there might lead to an overestimation.
Yeah, so to take that in reverse order, so the differences in high and low income countries, we would expect that the impact of reduction of bacterial transmission would have a greater impact in developing countries because the disease burden is higher. So carriage rates, or the rate at which children carry these organisms, tend to be higher in developing countries. Let's just use Africa as one example. So within Africa, the carriage rates are much higher than they are in the UK. So we would expect that the overall reduction in invasive disease would be greater in those countries as well by reducing transmission of pneumococcus or any of these. So we would expect a similar reduction, but even a greater reduction in developing countries. the first question was really about, if I can sort of paraphrase something more about the organisms themselves, which is part of the next phase of this work. So what we're reporting on at the moment are the cases and the dates of sampling, so when these cases arrived. But what we're also very interested in is how these disruptions have indeed changed the bacterial population structure, which is something that many of us think about for all sorts of different reasons. In other words, let's take pneumococcus as an example, that if you have indeed reduced the transmission of pneumococci and the natural transmission of that pneumococci between children and adults, then which pneumococcal organisms sort of survive that process and become, is there a change in the dominant types of pneumococci that circulate after what is effectively a bottleneck in transmission? So we're very interested in that. That we know has all sorts of really important issues. In other words, with respect to vaccination, we know that if you introduce a vaccine, say a pneumococcal vaccine, that disrupts the pneumococcal population and you get new strains emerging after that. So we might expect something similar to happen here. And the way that which we will assess that is not just simply by looking at cases, but actually looking at the characterization of those pneumococci. So which pneumococci are circulating afterwards. Some of that we can do now by knowing the serotype of these organisms. That's a polysaccharide capsule that surrounds the cell, and it's a way in which we characterize the organism. So we can assess changes in serotype. To be honest, at the moment, we haven't really seen any major changes in serotype. We've started looking at that. But the second phase of this will also be genomics, that we can start to look at which lineages of these organisms are circulating after this period of transition. I think the data that we've got so far suggests that we're not yet really out of this period of transition, that so far the data look pretty similar, but we will see. We won't be in these containment measures forever, and therefore the transmission will change, and we'll try to be in a position to be able to monitor that very quickly.
And do you think there might be a decrease in immunity to, say, sorry if I'm saying this wrong, pneumococcal infection in the future because of decreased exposure due to these different interventions?
No, it's a very good question, and that's entirely possible. If people are not exposed, they won't be developing the natural herd immunity. That's a sort of toxic phrase, but children will not be benefiting from being exposed to these organisms and developing natural immunity to those.
And we've had a question here, I think it sort of ties into what you were just discussing. Do you think it's feasible that the reservoir for these organisms has been fundamentally affected, in addition to sustained reduction in transmission?
I don't think the reservoir itself has changed. In other words, it's going to still reside in the same niche. But I think there's a very good possibility that what exists in that niche has changed. And the analogous example is indeed vaccination. So in almost every country that has introduced pneumococcal vaccine over the past 20 years, observes a change in the circulating pneumococci. And that's largely because you then vaccinate against a small fraction of the types of pneumococci. So the vaccines that exist now don't protect against all types, but they protect against a subset. So the subset of types reduces in prevalence, and then you see this emergence of types which are not in the vaccine, and sometimes those go on to cause disease. So the analogous scenario is indeed vaccination. And so I think we would be naive to think that there won't be some changes within the niche, but to what extent we see those changes, I think remains to be determined. And certainly that probably relates very much in part to how long we're sort of in this situation where you have reduced transmission. I would imagine that time is going to have a role to play in that as well.
Mm. And Hubert's just sent in that they published a paper last year in the BMJ proposing syndromic surveillance of influenza could be used to monitor COVID, as low middle income countries do have a pretty robust surveillance infrastructure on influenza. Given what you have found, do you think this should now be prioritized?
Sorry, what should be prioritized?
That's a good question.
The measurement of symptoms.
Surveillance infrastructure on influenza. Hubert, feel free to.
So I guess what sort of related to that, what countries have seen over the past year is a reduction in influenza infections as well. And so this was certainly the case whilst we were doing this first set of analyses, particularly places like Hong Kong. Margaret Ip runs the lab in Hong Kong, and she said that although they were having cases of influenza, it was nothing like what they would normally see in a typical year. The same was true for New Zealand. And typically they would have influenza cases before we would see them in, say, the UK or across Europe. So the containment measures certainly had an impact on more than just COVID, SARS-CoV-2, and certainly more than just these bacterial organisms, but probably on lots of respiratory transmitted organisms. What also is interesting is that I've seen recent discussions around increases in respiratory syncytial virus at the moment. This has apparently been true in parts of Europe and parts of America as well. Now, respiratory syncytial virus wouldn't normally circulate now. That's very odd. It would typically circulate in the months leading up to Christmas, sort of October, November, December time. So for it to be circulating now is unusual. So I think we have to be prepared for the fact that these changes that have occurred over the past 18 months or so will have a knock-on effect, and to what extent that has negative effects, as in increases in disease, remains to be seen. So I think we do need to watch closely. So I think in terms of surveillance, I certainly would advocate a close surveillance for the organisms that we're particularly concerned about.
Yeah, and sorry, just a correction, Hubert meant syndromic surveillance on flu. Yeah. So,
on flu, yeah.
Yeah.
I mean, that's trickier because you're talking about a respiratory virus, so the symptoms are often very, very similar amongst respiratory viruses. So,
yeah. Sarah's asking about, from the different containment measures for COVID, which do you think are most effective in preventing transmission of these invasive diseases?
I don't think we have the data to show that. So we could show that school closures didn't have a significant effect. We thought because these are organisms carried by young children that maybe the school closures were driving much of this, and that is not the case based on the first 6 months or first 5 months of last year. We didn't test any of the other measures independently, so I don't think we have data to support that. What we used is the stringency index as an overall index, and that contained all those different measures. So I don't think we have the data to support that. What we can say is that there was a difference in the relative stringency levels. In other words, some countries like the UK were in very strict containment measures, whereas others like Sweden and Finland and so on were in a more moderate level of stringency. And we can see differences there in stringency, but in our study, no difference in terms of invasive disease.
Mm. Angela, you're doing so well. The questions are just coming at you. So nearly there. Is there any evidence that COVID-19 is commonly followed by invasive bacterial disease, or do your data not suggest that this is the case?
So we can't answer that with the data we have. So it's data that we would be able to generate potentially going forward if the laboratories are able to look backwards and match up cases of invasive disease, let's just say invasive pneumococcal disease, with a diagnosis of COVID around that same period of time. At the moment, that was just more or less kind of impossible to do on the timescale that we were doing, because they have to get, each laboratory would have to get permission to do that. There have been anecdotal reports, and others might know more about whether this is starting to come out in the literature, but reports of subsequent infections, but nothing like what we were sort of fearing at the beginning of this IRIS study, that we might see this increase in invasive bacterial disease. We haven't seen that as far as we're aware. I don't think we're probably, sort of the things hiding in the data, as it were. So I don't think we've seen that. What I do know is that we're, in many cases, antibiotics were being prescribed for, like for example, azithromycin. The recovery trial was testing that as well, that azithromycin is being used to treat COVID patients, and these organisms would respond to azithromycin. So that may have actually reduced the incidence of subsequent invasive bacterial disease in patients with COVID if they were in fact receiving antibiotics, azithromycin otherwise.
It's fascinating. We've had a question from Toby asking whether older age at first colonization might affect the burden of invasive disease if transmission returns to pre-COVID levels.
Yeah, it's a good question. So in terms of colonization, if we just take a step back for a moment, Haemophilus and Pneumococcus behave in a pretty similar way. Neisseria also, as in they predominantly colonize children under the age of 5, and it's really in the early kind of first 2 or so years where the colonization rates are highest. For Meningococcus, it's slightly different. Certainly it is true for the younger age groups, but you can also have teenagers, for example, be colonized by Neisseria. That's why the vaccine is targeted at that age group as well. So if we just focus on the youngest age group, then you typically see that group being most highly colonized. And it's the period of time in their life where children then develop immunity to the different serotypes and the different types of pneumococci, or indeed Haemophilus, and that immunity will persist over the lifetime. The older age groups tend to have waning immunity to these organisms as well as to others, and of course they have other comorbid conditions in many cases. So a combination of both waning immunity and increased comorbidities mean that the disease burden goes up in that older age group. So the colonization rates of particularly pneumococcus and Haemophilus, and indeed meningococcus as well, tend to be quite low at the older age group. So you don't see many adults really colonized by these organisms. You can sometimes see parents of young children who are colonized, but that makes sense given that they're looking after children who are colonized. So the colonization rates in the older age groups tend to be much, much less than what you see in children. So are we going to see changes, I think is your question, are we going to see changes over time? I don't know is the answer. My guess is that probably isn't going to be the major driver of changes, because other aspects of that wouldn't necessarily be expected to change. In other words, the sort of path from childhood into adulthood would probably be the same. The immunity is almost certainly built up at those younger years, and therefore, as those children are exposed to fewer pneumococci, perhaps fewer Haemophilus, maybe that changes their immunity, and maybe that does change over time. But there are a lot of ifs there. I think we'll have to just wait to see what will happen.
It'll be really interesting to see what the data bring as the work progresses. And I think we're going to let you out of the hot seat at this point. You've done so well to receive all these questions and one by one get through them. So thank you so much. And really for a fantastic amount of work and a fantastic presentation to wrap up this term of Richard Dahl seminars. So thank you very much, Angela. And thank you very much to everyone who's here and to IT support, and enjoy the break, and we'll see you next term for more Richard Dahl seminars. Thank you.
Thank you, Karen. Thank you.